So I'm Ariel Alperstein. Like Erin said, I just started in the Department of Chemistry and Biochemistry a couple months ago. And I'm going to talk today a little bit about measuring protein secondary structures in tissue with two-dimensional infrared spectroscopy. And kind of the background I have in that and the extra concerns that we need to think about when we're doing this type of measurement and technique. And so the first thing that I want to do is kind of break down what I mean by this title and what I'm talking about, right? And so this core concept of my lab and what we do is, right, spectroscopy, right? Sending light to a sample and then measuring that light and getting spectral information from it. And specifically, I'm interested in vibrational spectroscopy, and today I'll be talking about infrared spectroscopy, but vibrational spectroscopy in general, right, measures intrinsic atomic motions along bonds. And when we're talking about proteins and protein secondary structure, what we're really talking about a lot of the times is this amide 1 mode. So here I have a little cartoon of part of a protein backbone, and an amide 1 mode would be this C double bond O stretching, and then a little bit of this NH kind of wag and bend at the same time. And that motion actually tells us about what structure the protein is in, because it's sensitive to the local environment, it's sensitive to hydrogen bonding, pH, other bonds around it, how hydrophobic or hydrophilic the environment is, and that all gives us information about protein secondary structure, and this is all commonly known, so you can go through the literature and see matches of crystal structures with this amide 1 peak and how it shifts and tells us about protein structure. And so, when we talk about measuring vibrational spectroscopy of protein structures, what we're really talking about is how can we measure how that changes with a disease state, right? So vibrational measurements of protein misfolding diseases can tell us a lot of information about how these protein structures might change over time. And so on the left here, I have a cartoon that I made thinking about how proteins aggregate in things like amyloid disease. So a protein or a peptide might have a native structure like the alpha helix on the left that gets disrupted. Maybe there's oxidative stress, maybe there's other forms of damage present, and that causes this structure to cleave, break down, or misfold in some way, and then it could potentially aggregate into a new structure known as an amyloid fibril. And there's been tons of studies measuring these structures using infrared spectroscopy in vitro in solution. So on the right here, I have an example of some TEM measurements on the left showing some native protein structures, and these are natively in 20 formers, so huge oligomers, of a protein called alpha B crystalline. That's a major component of the eye lens, but also found throughout the body as a protective chaperone protein. And a cartoon of that is on the bottom left. And then on the top right, I'm showing an aggregated structure of this protein. So there's a TEM showing that it's come out of these 20 formers in its native structure, and it's aggregated into these long, stringy fibrils. And on the bottom is an example of what some of those protein structures might look like in the fibrils. So we know that we can measure all of this in vitro and see all of these structures and think about how a particular protein might change in a disease state as the conditions around that protein change in the disease state. But one thing that's really important to remember when we do these measurements has to do with the title of this paper showing that structure on the bottom right. And that title is showing that fibrils in diseased tissue, right, protein structures in diseased tissue are quite different from these in vitro experiments. And so if we want to get a full picture of how protein structure might change during a disease, we need to think about how does what we see in the in vitro connect to what we see, ex vivo or in the tissue itself. And so some of the past work that I've done and some work that I hope to continue here is really trying to bridge the gap, measure both these in vitro experiments and the tissue experiments and try to understand what structures and what information we can take from each. And there's a lot of practical aspects to this, right? So we have to take tissue, we need to have it evaluated by people who might evaluate that particular disease. And so in this example, I'm showing eye lenses that were given to us as already with different cataract grades or as juvenile healthy tissues. Then we freeze them and we slice those using a cryomicrotome, right? Normal histology processes, except instead of putting on a normal glass side, we have to put these on IR transparent windows, so things like calcium fluoride. Again, for IR transparency, we need to remove any water, and we do that by, instead of adding a regular buffer on top of the sample, we add a deuterated buffer on top of the sample, and then we might add some kind of spacer, enclose this in a sample cell, and then collect images like this, where we kind of map over the tissue itself, and we get what the spectra on the left, which is a two-dimensional infrared spectra. So we're collecting, point map imaging of these different tissue slices to try to evaluate what's going on. And this experiment, for example, was about 25 micron thick tissue slices with a 50 micron spacer, so some level of buffer on top of that sample. Right, and so what is this two-dimensional infrared spectroscopy that we're using, right? So you might be familiar with Fourier transform infrared, which is what many people learn when they're starting out with general chemistry. And FTIR, Fourier transform infrared, right, generally is measuring these transitions from the ground vibrational state, that ground stretching motion, to the first vibrationally excited state. So now it has more energy. It might be stretching, at a different rate or a slightly different higher energy state. And when we do those measurements, we get, these peaks. We get what we think of as a normal spectrum. And that's what we generally think of with FTIR. With 2D-IR, in contrast, we send in some pump energy for that ground 0 to 1 transition, and then we send in our probe beam, which actually measures the transition from 1 to 0 and the transition from 1 to 2. And so we get a map now instead of just a single peak, and we can take diagonal slices of that map that look very similar to the peak on the left, but we get this multi-dimensional information now, very similar, if you know anything about NMR and 2D NMR, very similar to the increase in information content there. And why would we want to do this for tissue, right? We get a lot of benefits from doing this extra multidimensional technique as compared to the linear technique. And so one big part is we can actually pull out a small percentage of amyloid beta sheet secondary structure in a native beta sheet matrix. So in the eye lens, we have a ton of native beta sheet content. And if we're at a really small percentage of this amyloid beta sheet content, let's say less than 1%, we can't see that when we do the FTIR measurement due to the signal dependence being the transition dipole, this mu here, squared. And that transition dipole, if you remember back to when you took Gen Chem, thinking about Beer's law, A equals ELC, that E and that epsilon there is proportional to this mu squared. So it's an intrinsic thing about that particular bond that tells us the signal strength. But the signal dependence in 2D-IR for the diagonal is now that mu to the 4th. And more delocalized structures like amyloid beta sheets have higher mu values, higher transition dipole values, and so we can pull out those structures. So on the left here is an example of the exact same sample measured both ways, and you can really see that huge signal enhancement. So that's a huge benefit. And another benefit, right, is this spread along 2 axes. So right as we go from regular NMR to 2D NMR and we get an increase in information, we also get an increase in information when we go from FTIR to 2D-IR due to seeing features off the diagonal that can tell us more about the structures present or about how those structures are interplayed and related to each other, right? When we think about terms like coupling, how is this vibration, how is this bond over here coupled to this bond over here? We can see that come out in these off-diagonal features. And all of that together, right, this more sensitive, specific information is what helps us distinguish between the protein secondary structures in these tissue types, right? So here is an example of a scheme where the blue is juvenile lens tissue and the red is diagnosed cataract lens tissue, showing the examples of the 2D IR spectra at the top, and then in the bottom row showing on the left a diagonal slice, so a drastic difference between the diagonal slices. In the middle, a horizontal slice showing one pump frequency and all probe frequencies, and that's showing us differences in the actual peak width, thinking about anharmonicity of these vibrational modes. And in the bottom right, we're actually looking at these two cross peaks, this off-diagonal feature that tells us about the coupling overlaid, showing the differences between these two tissue types. So this is kind of an overlay scheme of how we can see the difference between protein structure in these two tissue types in one location. But right, we're measuring an image, we're getting, hundreds to thousands of pieces of information here. And so what can we do with that, right? So different ways of comparing these images helps us emphasize the difference between these different tissue types. And so on the left here, I'm showing an example of how we can compare some in vitro experiments to what's happening in the tissue. So in vitro, we had known amounts of different secondary structures combined. We made this little calibration curve. The blue sample is alpha B crystalline, and the purple sample is gamma D crystalline. And we can do this with different types of mixtures of proteins and different types of known structure. And then we compared that calibration curve to the tissue itself to be able to say the total percent of amyloid across the tissue type in general for this left figure. In the middle, right, this is a different way of thinking about and displaying this data. We can do an overall map and think about diagonal ratios or that cross peak, that coupling information, and make a map and literally show by color the difference between the protein structures and different tissue types. On the right here, it's actually some mouse pancreas tissue, and one thing we can do is think about how histograms of ratios change over time. So there's many different ways that we can kind of compare this image information to tell us something about the protein secondary structures and how they would relate to the different disease types of tissue we have. And so just to wrap up here, right, so future directions in the lab that I'd like to bring here at the University of Delaware are thinking about how can we make better spatial resolution and faster data acquisition. So just to go back real quick, right, you can see the spatial resolution for this center figure is quite pixely looking, and each of those boxes is about 100 microns. So not great spatial resolution, right? This is really an ensemble picture of the protein structures present, and we'll still be getting an ensemble picture with better resolution, but we may be able to distinguish between different regions of the tissue better. We want to do faster data acquisition so that we can get more detailed images and more of them with different types of tissue. We want to try to move to measurements in water instead of in deuterated buffer, and there's been some recent schemes within the past 3 years that have developed new techniques for thinking about how we can do these measurements in water and trying to lead to, thinking about how we can do cell and tissue imaging without having to worry about this water absorption effect. And then thinking about new tissue types that we haven't worked with and new problems. So this is an example of microplastics in mice liver and thinking about, measuring the protein structures close to those plastics versus slightly far away. And how does that structural information tell us about how much these plastics might be affecting the health of this tissue and the cells in the tissue? So thank you very much for listening to my short little introduction to this field today, and I'm happy to take any questions.